Today we are going to tissue culture. Tissue culture. So now students, tissue means a group of cells. Culture means medium which contains all the nutrients which necessary to grow the plants. In this process, in tissue culture, in nowadays or large scale use method is tissue culture, because in this method only some parts of plants used, like as leaves, roots or node, growing bud or node used mainly. So now in this process, take a piece of selective part of plants which we used to use in tissue culture, like as we use the part of root means the apical parts of roots, apical means apical meristematic tissue is present there, because regularly growth is going in this part. So now students, in this process, step first in this process, take a part of plants which have ability to multiply repeatedly, and then put into a test tube. Test tube which contains culture or medium. Okay, means test tube A. So now start the growth of part of plants. Okay, small pieces grow. Then second one is B. This test tube containing another medium or culture medium for the growing of plants, and then these pieces put into this medium, and the growing parts convert into undifferentiated parts. Undifferentiated, students, that means there is no differentiations, means which parts make to the aerial parts, pillimule, and which parts make radical, means rooted parts. It is a group of cell, undifferentiated group of cells. So now this structures inside this test tube is called callus. Callus means no differentiation. Then in this process, in this beaker contain medium which contains a medium. In this medium, plant hormones are present. So now when we put these callus in this beaker, and they develop as a new baby plants. New baby means a small structure only. A small means in this beaker we differentiate the part of the callus in upper parts to make a aerial parts or shoot part, and lower parts to form a root system. Then after that we put their small plants in soil or in a pot, and they grow as a mature plant. or transfer into the soil. Whole process is conducted inside the in vitro conditions, students, or we can say that sterile conditions. Sterile means inside the lab only. Sterile in vitro conditions only this process is conducting. Otherwise, not conducting the process outside and sterile conditions. Okay, students, this is the process of tissue culture. Very important, students, and completed into 3 steps, like first, second, and third step. Okay, students, how many steps? 3. Okay.